Okay, why don't we get started and then people will trickle in. We are very happy to have Adrienne Fairhall, who's with the University of Washington, and she's a professor at the Department of Physiology and Biophysics, to present the last talk of our symposium. And she's going to actually bridge the gap to behavior, and which is exciting. And so super happy to have Adrienne here. And she's done a lot of work on neural computation, and just personally, a lot of my thinking about how adaptations in neurons function have been very much influenced by her earlier papers. And so I'm particularly looking forward to hearing talk about how to take it beyond single neurons and actually bridge it to behavior. And so, Adrienne, please.
Thanks a lot. Yeah, thanks so much for the chance to be here. Great idea for a symposium, and it's a real pleasure. So this is a project that's a little bit different from other things we've done in my lab. So first, I'll just show you the cast of thousands who are involved. In particular, I'm going to be focusing on the work of my own student Hengji Wang and Josh Swore in this talk. But we got introduced to Hydra thanks to Rafa Yusta's lab at Columbia, and have had a lot of fun in the last couple of years learning about it and getting involved with that project. So Hydra, many of you might have come across them, but without knowing it. They live in fresh water, and they tend to hide kind of under leaves. They're about 1 millimeter in length, and they're cnidarians from the cnidarian family, so like jellyfish. So why are we interested in working on Hydra? One reason, it has a very simple nervous system. There's no ganglia or brain or really identified neurons. And so it's a bit different than another tiny system like C. elegans, where every neuron kind of has a name and a job and particular qualities that make it who it is. Hydra instead has a nerve net, and so is sort of a symmetric structure. And so that's a different kind of coding principle. And we were curious to understand how that works. At the same time, being very simple, it has a relatively complex behavioral repertoire. Unlike C. elegans, which kind of wriggles forward and wriggles backward, it functions in 3D, and so it can do complicated behaviors. Not that I'm going to tell you how it does those particular complicated behaviors, but in principle, there's the opportunity to build up to understanding a complicated behavioral repertoire. Another advantage is it's transparent, and so with the help of recent genetic lines, one can image directly activity in the neural net, the nerve net, and also in the muscles, which gives us the opportunity to really take the modeling kind of a step further, as I'll talk about today. Furthermore, it's been the subject of interest from Selena Giuliano's lab, who's done just a magnificent job in characterizing the cell types molecularly in Hydra. And so there's sort of a wealth of new information genetically about what's going on inside the cells, which we're just starting to scratch the surface of using. All right, so genetic lines, these with collaborations between Rob Steele and also in the Yusta lab, Christophe Dupre and John Savasky have worked on developing lines that allow you to do calcium imaging of the nerve net, and so that's what you see going on here. So this is what Hydra is just doing what it mainly does, which is stretch a bit and contract periodically. And so you can see there's flashing of distinct groups of neurons in the body. This is a really cool line, which they call watermelon, in which GCAMP is expressed, or GCAMP is expressed in the ectoderm, in the outer layer, and RCAMP is expressed in the inner layer. And these are expressions that are occurring in the muscle layers. And so now you're seeing the muscle activations. So it would be awesome. And they're trying to get lines in which you can see neural activity and muscle activity at the same time. But in the meantime, we have really a lot of beautiful, detailed kind of temporal information about these two aspects of behavior. Just to show you an example of the kinds of cool behaviors Hydra can do. So here is one somersaulting. So they tend to stick themselves to the ground on their peduncle, on their foot pad, and then pop off wave their tentacles around, search around on the ground, find somewhere to go and then pop their foot off, and they can kind of repeatedly do that and locomote around through this sort of somersaulting behavior. They also do another thing here, inchworming, where they kind of will crawl along the ground by making successive kind of grabs and movements. So we're not going to get to that because when we image, at the moment, most of the imaging is done in 2D. And so they have to be somewhat compressed between two cover slips in order to be able to hold them in place. But ultimately, we hope, and we're working with people who are planning to try to image Hydra in 3D using multiple cameras. So another aspect of their appeal is that they are, in principle, quite tractable for modeling. And modeling really, as New Yorkers say, kind of from soup to nuts, all the way up from the ground to behavior. They are essentially like a double layered bag, right? There's an outer layer of multivalent skin cells, which serve both as skin and as muscle. There's an inner layer of the same kind of cell. Those two layers are supported and held together by an acellular layer here called the mesoglia that gives the structure. And those two layers are separately innervated by nerve nets. So there's an inner nerve net, the endodermal nerve net, and an ectodermal nerve net. And what is kind of nice, and I think I won't talk about that. Yes, I will. So in the outer layer, the muscles are aligned longitudinally. And in the inner layer, the muscles are aligned circumferentially. And we'll talk a bit about how that leads to behavioral dynamics in a moment. So this is just to give you a quick, I'm not going to talk about this at all, but the sort of cell type map of Hydra that Selena Giuliano's lab has developed. So they've done a very detailed RNA-seq analysis while keeping track of the locations from which cells came in the body. So now are able to characterize the gene expression across the body and to find that there are quite a variety of different cell types. There are, I think, maybe 13 types of neurons in the nerve net. And in principle, we have access to all the genetic expression that goes along with those cell types. All right, so zoom out. So often when we think about understanding in neuroscience, this is a picture that at least I have always liked to think about, that you have some kind of complicated stuff going on in the environment, which you can characterize by the temporal evolution of a bunch of variables. You can record the neural activity, which is also complicated and time varying. And that generates somehow behavior, which is also complicated and time varying. And what we'd like to do ultimately is to reduce all of these high dimensional descriptions into some low dimensional set of latent variables, and to understand the relationships between them, how, what one says about the other, and how those all, what the sort of dynamical systems are that govern the evolution of these three properties. What I'm going to do here is take a little bit of a different perspective. Oh yeah, just to remind you, right, that we often leave out muscles from that picture, and that's an opportunity that we have here in Hydra. I'm going to take a different view here, which is that rather than at this point doing that kind of statistical analysis on observations, rather we're going to kind of take this more bottom-up perspective and see if we can just build a model basically from first principles that captures what the animal does. And so that turns out to really critically depend on dynamics in the muscle, which is not something we tend to take into account, I think, in general in neuroscience. We really generally think about neural activity to output, to behavior. But it turns out that in systems like this, the muscle dynamics themselves add a kind of additional layer of computation through their non-trivial dynamics. All right, so just to zoom in on my Hydra team, so this is going to be primarily work by Hengji and Josh, as I mentioned before, but we're getting some very helpful input from one of our visiting students, Shashank Sharma, and my colleague at UW, Madi Bosma, who's a gap junctional specialist. And we've also had Ethan Cato, who's a high schooler, kind of working in the lab as well, helping out. So one of the challenges in working with Hydra and trying to build that kind of statistical variant of the model is that it's not super easy to track the neurons. Part of the reason for that is that it is a flexible animal, and so it changes its aspect ratio by a factor of 4 to 10 as it stretches. And that is a challenge for a lot of the more machine learning approaches that people have applied over time. And so one of the things we've been collaborating both with Rafa's lab on and also doing a little bit on our own is to devise ways to analyze movement. I thought these were movies, but they're not. Okay. So we would like to have both characterizations of movement and detailed tracking of the individual neurons. And the way that we've decided to characterize the movement in our lab is to take the animal and to determine its contour, and we do that using an online package called IC. And then once we have the contour, we make use of deep lab cut to find a couple of points on the body. And those we decide to track, right? The peduncle, the very end of the body, the point here on the hippostome, and the two kind of armpits, where the tentacles meet the body. And with those 4 points, and that's kind of all we can track actually, because if those of you who have worked with deep lab cut know that you really need to focus on points that are identifiable by you, right, from frame to frame. And an animal like Hydra is quite symmetric, and there aren't really features on the body that one can easily identify from frame to frame. So we're kind of stuck with this rather low dimensional tracking using deep lab cut. But once we do this, along with the contour and those 4 points, what we can do is take away the tentacles, because they have a lot of activity going on, and we generally are not going to deal with them at this point. And we draw a line down the middle. We use the contour to kind of shave away the body and end up with a midline. And now we can track the shape and the stretch of that midline over time. Using the contour, we can mask and just calculate the total fluorescence in the body, and then compare that with the length. So here's just a typical trace where the blue line tells you the length of the body, and the green is the integrative fluorescence in the ectoderm. This is a neuron, an example where we're recording from neurons. We can use that information to do sort of maybe at this point, very simple-minded analysis. So we can find when the animal is contracting, when it's elongating, and when it's bending. And use that to turn the behavior into a time series. So now this is just some work that Thibaut Lagache in Rafa's lab has been working on for a while, and we recently have a preprint out about this. This is an effort to solve the tracking problem. And so here, what we're doing is finding spots and then watching them over time. One of the difficult aspects of Hydra right now, and again, this is a problem that's being worked on by Rafa and his team, is that we don't have neural markers. We don't have nuclear markers, right? So generally one would like to be able to see the neurons at all time, and that would make it a lot easier to find them. But when you only have information about the neurons when they're active and the animal's moving a lot, that gives us a tricky problem to solve, because one sort of needs to then join together the tracks that individual neurons create over time and deal with the fact that there's been a lot of distortion of the shape of the set of points. And so Thibaut has been working on methods to do best guesses at how to connect those tracks, and now is able to follow the animal over 500 to 1,000 frames, which is a big advance of what we had before. Without needing to completely solve that problem, there's a lot one can do. And given that one would also like to be able to analyze the movies where we don't have point-wise data, like for the neurons, but we're looking at, I think, so I'm going to assume that about 20% of these movies are not going to work. So I'll just jump past the ones that don't work. Let's see if that one works. Nope. Right, this is another example, I think, of neuron tracking. Yeah, so what I'm showing you here is a slightly alternate approach to solving the problem, where what we're doing is to use sort of state-of-the-art optic flow methods to try to estimate the movement of the animal, and basically to invert the optic flow so that we hold the animal fixed. If we were able to do that perfectly, then tracking is no big deal, because we can kind of move the animal into some reference location And now, even though it's moving, we can see it just as a stationary animal, and then we can just look at the neurons flashing over time. And the beauty of doing that is then we also have, kind of for free, a very highly resolved picture of the detailed movement of the animal, right? We have the complete flow field for all of the movements over time. So that's been what our lab has been focusing on. And if we have that flow field, the nice thing about that is now we don't really care about individual neurons necessarily. If we want to look at the muscle movies, we can just draw like a coordinate frame on the animal. And with respect to the stationarized animal, that will remain a constant coordinate frame. But now, if we then go back to the moving movie, that coordinate frame will kind of distort across the body. But now we can, say, integrate fluorescence in pieces of this frame, and that will give us the local fluorescence at the same time as this very highly resolved picture of movement. Right, so here's an example, I think, of, so here's the moving animal, and you can see this slow calcium wave moving through the body. And here's where we've mostly almost stationarized it, and now you can get a much better, we can do much better in characterizing that wave passing through the body because we've gotten rid of the movement. So I'd say this is still not perfect. We're still kind of working on it somewhat, but that's the approach that we're aiming to take in order to be able to do sort of the complete sort of statistical version of modeling that I started with. Right, so where we've started from in understanding neural activity is with this lovely paper from Christoph and Rafa in 2017. So Christoph, at this point, painstakingly by hand tracked neurons over some amount of time. And even that length of time allowed them to find a couple of interesting things. One is that the activity of many of the identifiable neurons in the body seems to fall into three gross patterns, right? So there's a whole bunch of neurons that fire synchronously that fall into one of these three groups. So rhythmic potential 1, contraction burst, and rhythmic potential 2. So these sort of subnetworks that seem to be almost entirely non-overlapping that are present in the body. Now, so just to point out, this RP1 and CB patterns both occur in the ectodermal nerve layer, and RP2 occurs in the endodermal layer. Now, what do we think that these neural networks might do? So just to remind you, Hydra is a hydrostatic skeleton. It encloses fluid in the interior, and it does that actually very tightly. There's some lovely work from Rob Steele showing that Hydra, because it's in fresh water, right, it absorbs water continuously, and so it will continue to kind of swell and swell. And from time to time, it has to ingest that fluid, and it also has to eat. And when it eats or ingests fluid, this hippostome actually has to rip itself open. That in between these periods, septate junctions reform across the barrier, making that a very tight seal. Right, so that says that there's fluid in this interior layer. As I told you before, the muscles in the outer layer are longitudinal, and the muscles in the interior are circumferential. So in order for Hydra to contract, what it needs to do, is to contract that outer layer of muscles, right? And presumably, when it wants to elongate again, what it should do is contract the circumferential layer so that will push the animal out again. That's typically how we think about a hydrodynamic skeleton working. You contract like this and you extend like that. But interestingly, what is seen in the data is a little bit different from that. So what is this one? This is an example of elongation. So you can see that there's clearly a lot of activation, right, of the muscle layers during contraction. And there are also slow behaviors that occur, right? So here's a few events where you can see, can you see my cursor here, my pointer?
Yeah. Okay,
great. Yeah, so you can see something getting triggered here that is gradually expanding, right? So there's a slow wave that's sort of growing out in the ectoderm that will often cause the animal to bend. And so here's another example where there's a bright patch down here at the peduncle, and that becomes the focus for where the animal bends. right? So there's sort of two time scales of muscle behavior, this very fast contraction and these slower, very local events that lead to bending. So what we wanted a model to do was to account for the data. You know, can we build a bottom-up model to simulate behaviors caused by specific neural activity? And there were a couple of puzzles that we wanted the model to solve. So one, how does one get these two different timescales of muscle dynamics? You know, typically when people model muscles, they model them as either being kind of having electrical signals through them, like cardiac muscle that have these very fast events, like we see during contraction, or they have slow events like peristalsis, like in the gut. But generally speaking, one doesn't have models, and we weren't able to find any where both of those dynamics were present in the same layer at the same time. So we wanted to figure out how those two could coexist. What is also very surprising, and I think I haven't shown you the data for this yet, but I will in a moment, both of the outer layer and the inner layer are activated during the contraction burst. So as we talk through, you know, how hydrostatic skeletons work, that is not what you would expect, right? You would only expect that the outer layer are activated during the contraction and that the inner layer, you know, potentially are activated during the elongation. What one sees, I think, no, sorry, I missed the slide where I have the fluorescence to show it, but it's somewhere down the track. What one sees is that there's fluorescent activity in both of the muscle layers that's very highly correlated during the contraction burst, so that they're both you know, the inner layer and the outer layer contracting at the same time, and there doesn't seem to be any muscle activity going on during the elongation. So we wanted to understand why that would be, what could account for that, and how Hydra can even contract when both the ectoderm and the endoderm are activated, because those two forces should be acting against one another. And so to solve this, what we did was to build a multi-layer model. So we start with a nerve net, and disappointingly for most of the talk, I'm not going to tell you about modeling neurons really. We're just going to take data that we get from the movies and use that to drive our models. But I'll talk a little bit, if I get time, on our thoughts about how the nerve net works a bit down the track. Really what we're going to be focusing on here is this epithelial network, is the dynamics within the muscle layers that can help us to account for the behavior and the dynamics that we see. And what we're going to add to that is a biomechanical model for the body to understand how these muscle layers and the active force that's represented by the calcium dynamics in these muscle layers then drives biomechanics, and therefore, you know, gives us a complete model of our little Hydra kind of puppet, basically. All right, and what that should ultimately allow one to do is to predict how neurons should fire in order to drive behavior, because we have the complete circuit from end to end. All right, so to start with, how do we get two timescales? And so we meaning Heng-Ji, you know, dived very deeply into the muscle literature and realized that one way to make this work is to put together parts that are known to be in many muscle cells, although many people that model them only look at one or the other, and that there are two well-known pathways for releasing calcium within muscle cells. So one is what we call a fast pathway that's mediated by electrical signals. So through voltage gated channels that then open voltage gated channels in the membrane and allow the flow of calcium in and out of the membrane, in and out of the cell. And there's also a slow pathway whereby a neuropeptide interacts with a G protein coupled receptor that then releases phosphorylates IP3, which then releases calcium from intracellular stores. And that is a slower process. G protein coupled receptors are generally acting on a slower timescale. And now what will happen to that the calcium that's generated through this, and also the IP3, which is kind of the messenger of this pathway, IP3 will diffuse across gap junctions, and calcium that's generated through a mechanism like this, the signals for that will propagate through the muscle sheet through diffusion of IP3. Whereas this fast pathway generates an electrical signal since we have gap junctions, those electrical signals will also be shared across the muscle sheet. And so we have two mechanisms that have very different timescales in terms of their activation dynamics within the cell. And they also have very different timescales in terms of their propagation. The electrical signal propagates rapidly, right? Because it's just it's a voltage signal that's being shared across cells. Whereas this slower signal is going to propagate diffusively through the movement of IP3 from cell to cell. All right. Something else I want to mention, but that's okay. Okay, so now let's just put that into the model and make sure it works. So we can zoom in on a Hydra, we can count all the cells, and we can build a model that's about the same size, right? It has about 60 by 30 cells, consistent with our counting. We build these two layers, and we make each cell in the network. We give it these dynamics, and not surprisingly, we can reproduce these body column waves, right? They can be triggered by the activation of just a few muscle cells. So we assume that maybe there's one neuron that's sort of speaking to a little local cluster of cells. It does seem to need more than one to be activated in order for these waves to be triggered. Oh yeah, the thing I wanted to point out was that it seems to be key, at least in our hands, for those two pathways to be triggered by distinct inputs, that they really are driven through potentially by neuropeptides or some form of neuromuscular coupling that is different for the two. And so that is great, right? Because it tells us that there are likely sort of different control pathways that neurons have in order to communicate with the muscle layers to drive these different dynamics. All right, so we can reproduce body column waves that look very much like they do in the animal. We can reproduce these bending waves by triggering a little group of cells down near the peduncle. And we can get the fast waves by triggering the electrical pathway. And because data has suggested, over many years of recording from a bunch of labs, that the electrical wave propagates from peduncle to hippostome, that is how we assume that they get triggered. So we started by triggering just down here in the peduncle. What I didn't show you, but is the case, and you might actually remember, though it flashed by very quickly, the neurons that are in the contraction burst network of neurons are actually distributed throughout the body. So there's not a priori a good reason that we would only trigger them from the peduncle, because there is a network of neurons that's fully distributed that does light up at the time of the contraction burst. But because the data tell us that the wave of contraction seems to originate from the peduncle, the way we've chosen to drive them is by assuming that only the neurons in the peduncle are important for triggering that wave to occur.
Can I ask a question, Adrienne? So I was kind of curious, in the particular mechanism you showed, like how much of a separation of the two timescales can you achieve, and like how much do you need?
Right. Yeah. They are well separated in timescale. The question is, can they possibly interfere with one another? And if you have a fast wave going through, does that sort of take up all the calcium and then not allow the slow pathway to occur, for example? And there is a little bit of interference, but there's also parameters in the modeling that can make those two pathways more or less independent. So we have assumed that they are fairly independent, but it would be definitely an important thing to check that everything we see in the data is consistent with that. Certainly it's we see in the data that when the animal contracts, it can also then begin a bending wave that, as it now uncontracts, it ends up bending in the right direction. So the two dynamics are able to overlay. We know that from the data. And so our model, we chose to reproduce that, but it would be good, just to test that we've done the right thing, that it's not too sensitive to that choice. All right, so I've shown you that we can construct a model that accounts for those two timescales. Why are both layers activated during the CB? So during the contraction burst, I mentioned to you that both the muscle layers, the internal and the external muscle layers, are both activated. They both have fluorescence in them during the contraction burst. But the CB network, the neural net, the nerve net that fires that, is only in the ectoderm. And there has been no evidence, and people have looked from EM microscopy that there's any connection between the neurons of the two layers. And we just don't see any activation of neurons in the endoderm during the contraction burst. And so this is, it's a mystery, but not a very, deep mystery. And that one easy way to account for that is that, oh yeah, finally, this is a slide I've been wanting. All right, so this shows you the activation of the calcium in the ectoderm and in the endoderm during these cycles of contraction and elongation. And you can see that the ectoderm and the endoderm are co-activated and that they decay away. And then during this cycle of elongation, there's essentially no calcium activation in either layer. So why is that? One simple proposal for that is that the two muscle layers are coupled so that the coupling doesn't occur through connections between the neurons that drive the muscles, but instead the muscle layers may be directly coupled. And there is EM evidence for gap junctions that actually cross over the mesoglia that could account for that coupling. Now, you don't want too much coupling because the electrical activity that causes the contraction burst is shared between the two layers, but these slow activations that cause bending are not shared. You only ever see that in the ectoderm. And so one can we showed that if you vary the coupling ratio, then you can find a kind of sweet spot whereby the coupling is strong enough and dense enough that fast waves will be shared across the two layers. So here's the ectoderm and here's the endoderm. So you get co-activation in the two, even though the neural drive is only in the ectoderm, whereas the slow wave only occurs in the outer layer and not in the inner layer. If you make it too dense, you do get leak, of that slow activation across the layers. If you make it too weak, then you won't get the inner layer to drive. So small mystery, but it does seem to be well accounted for by the postulation of an intermediate level of gap junctional coupling between the two layers. All right, so the last question to answer here is how can Hydra contract when both the ectoderm and the endoderm are activated? And to answer that, we need to move to the final step here of dealing with the biomechanics of the body. All right, so everything I've told you about so far is about the calcium dynamics in these two epithelial networks. Calcium translates to active stress by the way it drives the myosin dynamics in the muscle layers. And so we can take our calcium, convert it into stress, and turn that into an active drive into our biomechanics. And the way we do that is through a well-known model, the High-Murphy model, which deals with the dynamics of cross-bridge coupling in the myosin layer. Won't go into detail about that. We're just taking that as a standard model. We do need to fit the parameters of that model to tell us, and what this gives for us is the relationship between calcium and stress. And then to do the full-on biomechanical model, we resort to COMSOL multiphysics, which is a package that lets you build a finite element model. So we have a finite element model of the Hydra shell. We've abstracted it down to a simplified version like this, a kind of cylinder with closed ends. We take each of the elements in the finite element model to have viscoelastic properties with, they need sort of turns out to have this sort of hyperelastic property, which is a relationship between stress and strain that's typically observed in various kinds of biological tissues. So that all of this takes, sorry, Stephen, go ahead.
Yeah, so quickly, this hydro or hyperelastic dependency, that's got essentially a bistability associated with it, correct?
Not in our hands. I think that the damping is enough that is not going to lead to, it's really a very damped system. And so all this tells you is just, it's not simply a linear relationship. It's a little bit nonlinear, but I don't think there's any.
The signature that I'm looking at is the lack of convexity. So that's basically the signature of bistability. We can look at this at the end of the talk. I don't want to hold you
Yeah,
up.
right, right.
That's a very interesting constitutive law.
Right. Yeah, I don't think one can ever get this guy into a regime in which it oscillates or does anything complicated. It's just very, very damped. And so I don't think the fact that there is a slight nonlinearity to that curve is going to cause any
I think there's actually from a mathematical standpoint, there's something very interesting about that particular feature. So
yeah, no, I get what you mean, from the shape of the curve, but I'm not sure we're ever in a regime where we will explore anything.
Well, I mean, if the strain goes beyond the point of inflection, then indeed you're in the regime that's of interest.
Yeah. We can come back to that. I'm not sure I'll have anything very smart to say about it, but yeah, it's an interesting point. Thank you. All right, so this is our Hydra. We've given it passive viscoelastic properties. Thanks to COMSOL, we can deal with fluid in the interior. That's very important, obviously, because the fluid is what transmits pressure, right? So as the animal contracts, then you have to distribute that pressure over the rest of the body, and so COMSOL fortunately takes care of all that distribution and all the boundary conditions for us and the properties of the interior water. And to that, we then add, so here's what it looks like. So if we were just to take that model and drive it the way one imagines that a hydrostatic skeleton should work, if you squeeze the endodermal muscles, you get stretch, and if you squeeze the ectodermal muscles, you get it to shrink, just as you'd like to have happen. So what happens if we take those two muscle layers and drive them the way we see in the fluorescence, right? And so now you see exactly what you would worry about, is that the contraction from the longitudinal muscles is fighting against the contraction from the endodermal muscles, which are kind of moving in opposite directions, right? Which are pushing the fluid around in opposite directions, and you can't get the contraction that you see in the real animal. So how did we resolve this? Hengji had the smart thought that what might be going on, and one way to account for that is that the properties of the muscle layers in the interior, in the endoderm and the ectoderm, might be different. Right? This high Murphy model, as I said, has a lot of parameters, and there are ways to set them up, right? So you can sort of dial the muscle properties between what's called phasic, so that has very rapidly turning on and turning off, or you can form these sort of cross bridges by turning down you can sort of stick the muscle into a stretched, a contracted state, and hold it there for longer by changing these time constants of moving in and out of this sort of cross-bridge state. And so what that does is makes the muscle kind of contract and then just sort of hang out there, to stay contracted for much longer. And so if we assume that the two muscle layers have these different properties, that helps us to account for both things, right? So now we understand that the endoderm can quickly cause the animal to contract. The endoderm will respond more slowly, but then once it starts to respond, it will stay on, and that accounts without needing to have any additional fluorescence accounting for kind of the active elongation at the end. It was sort of mysterious that the animal could elongate without apparently needing to drive those circumferential muscles. But these differential muscle dynamics allow that to happen. All right, so that explains both how the animal can contract despite sort of the contra action of the interior and exterior muscles, and how you get this extended elongation without any apparent fluorescence. All right, so putting that all together, what we've addressed, we've understood how we can account for the two timescales of muscle dynamics, why both layers are activated during the contraction burst, and how Hydra can contract when both the endoderm and the ectoderm are activated. So now what we'd like to do is to take our puppet Hydra and drive it with neural activity to see if we get anything like real behavior. So we can go back to the calcium signals from the neural animals and extract the contraction burst events. And we're also going to add into that these red lines, which are the activations of bending, right? So we don't see them directly at the moment in the neuronal data, and that's possibly because if they only fire once or twice, then calcium might not pick that up. But if they're also buried in the very intense fluorescence of the contraction burst, they just may not be visible. But we also don't have perfect cell tracking, through enough cycles maybe to be able to find them. Anyway, we assume they're there because we see Hydra bending. So we pull out sort of moments of initiation of bending waves. We convert that into, we drive that through our muscle layers. We generate the calcium signals, which then turns into force that we put into our biomechanical model. And now we get something like this. So as the neurons firing, first the CB network will cause the animal to contract, and then those bending waves, we're generating them at different locations around the peduncle, and they will bend in the appropriate direction. So we get pretty realistic Hydra looking behavior out of the model. And now if we compare it, I think I don't want to say quantitatively, but somewhat quantitatively, we now compare the length of the animal with the length that we extract through our imaging. We can find parameters that fit really well, right? So we can kind of reproduce these complete cycles of elongation and contraction, where we're able to account through our viscoelastic properties for that slight bouncing in the length that happens during the contraction, and also the timescales of expansion. And often when we get deviations, if you look in the movies, you see that the animal has done something like use its tentacles and either slow itself down or kind of stretch itself out a little bit more than the model would predict. So we haven't taken into account kind of the stickiness and the extra help that the tentacles can give it in the behavior. And just to show you, here's another example. So this is one where it was sort of sticking itself by tentacles. So there's a bit of a deviation in that elongation. It's sort of stopping itself from elongating by holding on. But otherwise, without any changes in parameters, we're getting a pretty good fit to that second one. All right, to summarize, we've developed this bottom-up model to simulate behaviors caused by specific neural activity, and have addressed these puzzles by postulating that there are two different signaling pathways that are likely independently controlled through different neuropeptide signals. We see different mechanisms that establish typical length scales and the effective dimensionality and dynamics of the control space, and that our model shows model suggests that one way to account for behavior despite what we're observing in the fluorescence is that those muscle layers have different properties in terms of their relationship between calcium and force generation. Okay, any questions about that? Because I'm going to, oh yeah, no, there's one last thing I want to show you, which is what we're working on now. So obviously one question that one might, as a reviewer of our paper, have is, can one do this? Can one think about this in a simpler way, right? So what we've done really at this point is really a full-on simulation. So we have every cell has a lot of parameters that we've had to choose. We have to choose how they're coupled with one another. Can we abstract a little bit from that level of precision and detail to a slightly kind of lower dimensional model? And a very natural way to do that, I think, is to extract kind of the fast and slow kernels, right, of the calcium and voltage. And so certainly one can through simulations, right? So here's just basically the point spread function, right, of what calcium and voltages are doing when they are triggered in a single muscle cell. And I think the next one, can I go on? Oh yeah, here's the slow kernel. So these are what the slow waves are doing, the calcium and the voltage. So at least numerically, we can extract a spatiotemporal kernel that is the response of the muscle layer to a single neural activation. One would like to test and make sure or find out whether thinking about them as a linear kernel is correct, and they're not quite linear, right? So here's 2 points of stimulation of the fast dynamics. And let's wait for that to go through again. Yeah, so you can see that they don't just sum, right? There is really interference at the points where those two activation events meet up. And that's also true in slow dynamics, which we shouldn't really be surprised about, right? There's a refractoriness in the dynamics that's going to lead to them not being able to be activated multiple times rapidly. And so here you can see that similar kind of cancellation of events as they contact one another. And this is something that was also discussed in some really nice recent modeling in jellyfish by the Memesheimer lab. So yeah, so one way that we might have expected to see that. So as you remember, when we triggered our fast wave dynamics, we did that entirely from the peduncle. One might think that we should be able to just see by eye whether because these nonlinear summations, whether that's the right model or not by looking at the activation patterns across the sheet after the beginning of a contraction burst. But because if it were being driven by this distributed contraction burst network, then one might expect to see these kind of edges as you're seeing here in the simulation. This is actually for the slow dynamics, but the fast dynamics has some similar effects. But I'm not sure that we have the resolution to see that, because once the contraction burst goes, the activation gets very large and kind of and the animal is all contracted. So if there were kind of edges in those dynamics in the calcium activations, I'm not sure we would be able to resolve them, unfortunately. Okay, that's it for muscles and their behavior. And I'm just going to spend a little bit of time telling you where we're at in thinking about the neural network, which I would say we haven't solved it and haven't figured it out yet, but we have some thoughts about how to build that network and how to make it do some of the things at least we're seeing in the data. So if there are any questions about what we've talked about so far, it'd be a great time to take them. Anyway, I'll just stop talking for a moment, which is good.
There is something in the chat, kind of broad.
I'm not seeing the chat.
It's about how Hydra sense and actively search for food, and if it's understood.
Right. Yeah. No, those are great questions, and not really part of the studies that we've done so far. So Hydra has an old history. There's a lot of people have been interested in Hydra since Tremblay in the 1700s, and so there are a bunch, long kind of track of older lovely papers that have done various kinds of qualitative observations about Hydra. So a few things are known. Hydra tends to elongate in the light. It has some kind of chemotaxis, or it does seem to move toward odor sources, but primarily what we see just by watching the animal is that it tends to eat when it physically contacts a creature, and so that when it touches an animal, that triggers sensory neurons in the tentacles that then shoot a nematocele, these little kind of poison dots, which jellyfish also have, into the animal, and then it kind of stuffs its food into this newly torn mouth. And so all of that we are not dealing with at this point. We really don't know very much systematically about the sensory responses of neurons. And that's sort of a new area, because people previously have made measurements, but it's very, very hard to patch Hydra neurons. And every year at Woods Hole, all the Drosophila guys think they're going to solve this for us, and they all try it, and so far no one's really done it successfully. So patching individual neurons and figuring out what's going on inside them has been very difficult. So these new genetic lines are a real they're a kind of a new threshold, I'd say, for Hydra. And one of the people who's doing sort of maybe some beautiful systematic work on this at the moment is Jacob Robinson at Rice. And so what Jacob is doing is holding the Hydra in sort of microfluidic little chambers, so that he can very carefully control both say temperature and properties of the external medium while being able to record and image the neural network by sort of holding them fixed and being able to do sort of systematic variations. Rafa's lab are also doing related things, and I'll show you a little bit of data from that in a moment. But we're sort of just at the threshold of really figuring out sort of the sensory responses of Hydra, even though much has been written about it over time, but more from a behavioral point of view. All right, so back to what we know with a bit more precision about Hydra's neural activity. As I told you before, there are these three subnetworks that have these sort of large-scale synchronous patterns of activation. One of them is very strongly connected with behavior. That's this contraction burst. The other two, I'd say, are a lot less strongly, obviously connected. There is no clear instantaneous behavioral response to either of these events in the neural activity, and so there may be slower timescales of mediation of what they do, but that's not at the moment exactly known. So we have been trying to understand the contraction network, what's going on there, and one proposal that's sort of emerging from combination of things we've been seeing and also from Jacob's lab is that one may be able to think about this as sort of being a distributed sensory network that's sort of getting inputs in a distributed way across the network, and that it seems that the hippostome is very important to kind of collecting that signal and potentially activating contraction bursts, because if you cut off the hippostome, then you really diminish the contraction burst. But it also seems very clear, and this is sort of obvious when you watch Hydra, is that the main event of the contraction burst is down here in the peduncle, and that's why we modeled the muscle activations as kind of coming from that. So there's a very tightly connected synchronous part of the contraction burst that's down here in the peduncle, and one possibility is that the peduncle is kind of acting as an amplification network of the activation that's coming from the hippostome. So one possibility is that the network is kind of collecting information, activates up here at the hippostome, that then drives a big wave of amplification at the peduncle. So, we'd like to understand what's sort of the pacemaker, right? This activation of contraction burst has sort of two timescales. On every contraction burst, there's sort of this rapid sequence of pulses that is a given contraction event, and they reoccur somewhat periodically. And so these guys, Charles David and collaborators, some years ago, tried to understand whether the fact that neurons in the peduncle seem to be heavily gap junctionally connected might be a contributor to the pacemaker-like nature of in CB. And so what's the evidence for that? So here's in control, what you're seeing here are the contraction burst events. So the red line is the initiation, the gray is the extent of the contraction burst events. So they kind of chug away quasi-periodically. If you now put a anti-INX, so you block, I guess, the gap junctions. So down here in the peduncle, so what you're seeing in blue are a particular type of innexin. So Hydra has a bunch of innexins, so gap junctions are obviously very important within Hydra, and neurons in the CB seem to have these innexin gap junctions sort of primarily down here in the peduncle. And so if you block innexin 2, then that drops the rate of contractions by a lot. You can also apply heptanol, which is known to block gap junctions, and that drops it even more. People who work on gap junctions don't like this experiment very much because heptanol is a very dirty drug and has a lot of other influences, but it's at least suggestive that gap junctional coupling is important for maintaining the rate of contractions. The idea that the peduncle itself might be the pacemaker is a little bit belied, I'd say, by data like this. This is also data from Charlie David, where he just cut a hydra in 2 pieces. And if you look at what's going on here in the peduncle, it doesn't move very much at all, whereas the hippostome is super active. And so the initial idea that maybe this very intense gap junctional coupling leads to it being sort of super excitable and thus being able to act as the pacemaker isn't really borne out by data like this. And so Josh in our lab has repeated this experiment now with the GCamp animals. And first, just to try and see if we reproduce that finding. So here is the peduncular half, and here's the hippostomal half. And we see something very similar, which is that the hippostomal half is much, much more active. And you can see here these bursts of neuronal firing. And so that, and so when we look at many examples of these dynamics, the number of contractions per hour is much lower in the peduncular half than it is in the hippostomal half. Something that's neat about Hydra is that it's one of these regenerative animals. It's very, very regenerative. You can cut into many pieces, and every piece will grow itself back into a Hydra. You'll work from Jonathan Lovas in Rafa's lab. He's been working on going the whole hog and actually dissociating the entire animal and then centrifuging it back into a ball, and you find that ball can build itself back into a Hydra. So it's really good at putting itself back together again and growing a new one. And so we can watch as the animal kind of recovers itself and both halves grow back, and then you see that you recover that original or the kind of native frequency of contraction bursts over time in the 2 halves. All right, so this sort of takes us back to this idea of whether that the activation network might be up here in the tentacles, and maybe you could even see that as the tentacular half was kind of bouncing around, there seemed to be a lot of sensory activation coming into the tentacles that maybe was driving neural activity there, that may be then integrated over time in order to drive the contractions to happen. That's one possibility. So, the picture is still a little vague, but we'd like to try to understand how this network, which at first pass we assume is primarily gap junctionally coupled. There's some EM evidence for synaptic connections, but given that the networks tend to fire very synchronously over the entire body, I think it's a good start to assume that they're primarily gap junctionally coupled. So these contraction burst neurons fire almost simultaneously. There are this second network of neurons, the RP network, which fires in this sort of clockwork-like way throughout time and doesn't have an immediate effect on behavior, but has a slight anti-correlation with the CB network. So might be acting as kind of a slow inhibitory input into that network. So what we'd like to understand is where these sort of 2 timescales of behavior are coming from. The short timescale that generates the rapid sequence of bursts. There's some possible evidence, just looking at some of Jonathan's data, when he dissociates an animal, you do see some cells that continue to fire kind of rapid pulses. And so that rapid firing might possibly be an intrinsic property. What we haven't done is to sort of track that over time as those neurons get sort of reintegrated back into the network, whether that they remain they hold that pacemaker-like property, or whether the ones that have that rapid firing tend to integrate themselves into specific locations within the network. Whereas this long time scale behavior is possibly partly due to a kind of seesaw interaction between the CB network and the RP network. But what we're currently wondering about is the contribution of potentially of sensory inputs that are coming about through mechanosensation, although again, we have no direct evidence that there are neurons that are reporting stretch, right? But another possibility is that the contribution of water might be important. So what contraction bursts are probably doing, and I think, you know, some of the things we talked about already are kind of pointing at this. Remember I told you that Hydra is a freshwater animal, and so it's going to be constantly absorbing water. One possibility for the contraction burst, for why both muscle layers are acting at the same time, is that might be a mechanism to almost sort of squeeze out water, right? So as the tissue is kind of swelling, then from time to time, you kind of run both muscle layers together and you kind of wring out water, which will be, you know, both go to the exterior of the animal and also go into the interior. And then after many of these contraction burst cycles, the animal has to expel the water, and then it does this sort of adjustion event and gets back to normal. And so it's possible that the water, the swelling by the absorption of water, may itself provide the control signal that causes the cycle of contraction. So we need to do some back of the envelope calculations, which we haven't yet, as to whether it's plausible that osmotic forces could lead to enough of a change in the volume of the cells to lead to a change in the ionic concentrations that would change the rest potential, right, of the animal. But certainly there are models around that would predict that as one changes, you know, the effective V rest due to that changing ionic concentration, that could lead to a slow oscillation. And we know that osmolarity matters. So there's another paper from Rafa's lab from last year where they just varied the properties of the, you know, so the salt properties of the exterior medium, and show that as a function of osmolarity, you do change the frequency of the contraction burst events. So we know that has an effect on the contraction bursts. So that seems like, you know, that's at least a kind of a first pass possibility of what might be driving the slow timescales, and we're currently building a model, you know, trying to see if we can get all of that right. So one of the pieces that we'd like to put in, you know, based on this genetic information is, since it seems clear, right, from the distribution of innexin 2, that different locations within the body have very different gap junctional coupling properties that may be very important for the function of the animal. And so here, you know, here's a zoom in on the gene expression in the different neuron types that were identified by Selena's lab. And you can see that, you know, there are many types of innexins. So there are these 5 different types that are expressed in neurons, and they are expressed differentially across the body. So what we've been doing is to look in detail at the properties of those innexins, because they may establish a lot of the dynamics of the network. And so Josh has been working with N2A cells, which themselves don't couple together, and expressing in them different kinds of innexins. And so once they have the innexins expressed in them, then they actually do form gap junctions. And now we can use them as a test bed to test the physiological properties of those different gap junctional connections. And so, yeah, so just to demonstrate, those cells themselves will not intrinsically couple. Nothing ever happens. But if they express gap junctions that are functional, then now if you put an electrode in the 2 cells, you can drive one of them and you can record from the other one and just test the properties of that connection. And that's important to do because, you know, these gene expression scatter plots are awesome, and we know that these you know, there's the potential for expression of the gap junctions, but we don't know without doing this kind of physiology whether they do in fact form functional connections. And so here's just a couple of examples of data from looking at the coupling properties. So here, Josh injects current at different steps, and you see that they do actually inactivate. So, you know, I think as modelers, we often think about gap junctions as just kind of static things that just cause an electrical, you know, lead to just a continuous pure electrical coupling, but in fact they do have dynamics that we do need to take into account. So here they inactivate, and they don't have completely linear current voltage relationships. So they inactivate, and now you get these kind of nonlinear relationships between voltage and the final current that's able to be transmitted. And so Josh is going through very carefully with each of these inexon types and characterizing their behaviors. We can see single channel conductances, so something I also didn't realize you could see with gap junction. And so you can actually watch them fluctuating open and closed as you can for voltage dependent channels. And so from this data, we're able to extract the gap junctional coupling coefficient, which is pretty nice. And that's, this is traces from a few different channel types. Yeah, so able to figure out the single channel current that is driven through these different inexon channels. They also have distinct timescales of activation. So when you switch them on, they have different rise times. Again, in the models, and in Hangji's model in particular for the muscle layers, we'd been assuming that voltage is shared basically instantaneously across the muscle sheet, but this data, these are all neural gap junctions, so Josh hasn't, these are a lot of work, all of this data, so hasn't yet looked at the muscle ones, but we may need to think about the dynamics of the propagation of the electrical signals as being limited by the dynamics of gap junctions also. So this really should be done as well for the inexons that we think are at play in the muscle layers. Final question that's important, I think, to ask about gap junctions is whether they can form heterotypic junctions. So at the moment, we've just been looking at whether each individual type of inexon is able to form a functional junction. There's also the possibility that you can get gap junctions between different inexons, and that would lead to the possibility of thinking about sort of a connectome, right, from the different cell types that were identified through the genetic analysis. It could be that there are specific ways that neural groups with different inexons may be able to talk to one another. If they all have the same inexon and you only can have you can only be gap junctionally coupled with the same inexon, then that would explain, right, that the gene type data would sort of explain these sort of functional groups, and one may just be able to tell, right, who's in which network by which inexon they're talking to one another in. But if they are heterotypic, that gives the opportunity for different groups to talk to one another. And it also, a known property of heterotypic gap junctions is that they can be associated with rectification, which would allow information to only travel in one direction, which would be something one would like to know about. So haven't yet, Josh has tried a couple of different combinations, but we haven't yet found any heterotypic junctions that work. All right, so that's where I'm going to stop. So I've been quite excited about thinking about Hydra as sort of a test bed for systems neuroscience, because we have this opportunity to really go from the ground up, from expression of inexons to figure out the properties of the underlying sort of molecular dynamics that lead to the behavior of the neural groups, and then we transform that into muscle activity and then into behavior. So you will have noticed that there are a lot of things we had to make up along the way, and I think that's where the modeling has actually been helpful, right? Because we had sort of conundrums that we needed to assume particular properties of the muscle dynamics and of the body dynamics that lead us to make predictions about what the physiology is going to turn up, and they stand as sort of predictions that are yet to be validated. Yeah, so what's left? So obviously we've barely scratched the surface. We're dealing with a very simple behavior. Contraction is just a whole body simple thing. We don't yet really understand the control of that one simple thing in terms of the oscillation, but we have some ideas about the feedback mechanisms that might lead to that. I think what's more exciting down the track is to try to understand how this little creature, with no central sort of decision-making nodes, right, carries out sort of purposeful behavior. When you watch it somersaulting in a particular direction, how is it that a network of perhaps essentially identical neurons can sort of organize themselves to do something that ends up looking purposeful? And so I think that's where this is a kind of a fun model for the future, is how does purposeful behavior, right, emerge from this sort of distributed computation. Okay, I think, yeah, just back to my thank you everybody slide. So we'll leave it at that, take questions.
Okay, let's all thank the speaker. Adrienne, that was wonderful. So thank you very much. And I had a question. So what might be sort of the most, if you were to think about the most complex behavior Hydra exhibits, which I don't know, yeah, which might be tractable in some quantitative way, what might that be? And
yeah, so I guess somersaulting is the dream, right? That's what we'd like to understand.